So forgive some of our group not being here. There's actually a big meeting in New York that a lot of the co-PIs and the leadership team are at. So it's tough when you have competing meetings.
Sandy, did you request that? Oh, there it is. It's recording. Okay. Each talk will be back to back, approximately 20 minutes each, followed by a short Q&A for both presenters. Lastly, additional information about future ATE project talks can be found on the Innovate Bio website, which we will put in the chat. Thank you. So it is with great pleasure that I introduce our first project talk speakers. From Southeast Community College in Lincoln, Nebraska, Misty Wheeling is the PI, along with Dr. Tracy Nadai and Gabrielle Johnson. Their presentation is titled Social Dogs. Their webinar will focus on implementation of an ATE project centered on connecting a dog's genotype to their phenotypic behavior, helping to answer the question of why dogs are much more social than wolves. Welcome.
Okay, hopefully I can share my screen here. Looks like I can. Okay.
I bet you're all dog owners out there, right?
Mm.
I have 3.
3 dogs. Cool. Okay, so we are in the 2nd year of our ATE project. And so we're gonna give an overview of where we're from and what we're doing, and at the end connect you to the resources that have been built so far, as well as update you on how many people are getting involved. So we are from Southeast Community College, which has a 15-county service area in Southeast Nebraska. And so that's been our primary area of focus, but we have done some sharing at High Tech, and we're hoping that more community college instructors will get involved as well. And our program is a relatively new program. Our program at our community college officially started in the fall of 2021, but we've been working towards that goal for even longer. And we were able to receive some funding from Innovate Bio that helped us pilot some of the stuff that became part of our ATE proposal and now our project. So we were able to apply and receive Innovate Bio's grant that they were doing for implementing undergraduate research with students. And that was focused on community college students. At the same time, we were also getting involved with our industry partner that is on this project, and starting to work with high school teachers to increase their awareness of biotech. And then that project grew out of that. The very first workshop that we hosted for teachers, we learned a lot from the teachers about their needs, and that led us to then applying for and receiving this ATE grant. And just a little bit about our program. It is a series of 3 stackable credentials that students can complete. And after any of the 3 credentials, certificate, diploma, or associate of science degree, the goal is that they're able and ready to enter the workforce. And they can also transfer. We're working on even more articulation agreements with the University of Nebraska and other local colleges. We've also had some students with bachelor's degrees come back and get a certificate so that then they're able to more easily enter the workforce. Okay.
Okay, so I will talk a little bit about our goals of this ATE project. So we have our different aims here to increase awareness of biotechnology at the secondary level. We see this as a great way to provide students with an opportunity that they're really personally invested in, really excited about, and maybe they will then decide to pursue biotechnology as a career. So within this ATE project, we have kind of two projects. We're calling them course-based research experiences or cures. The first project is our canine genetics project that we'll mostly be talking about today. So we are reaching out and working with high school ag students, biology students. We also do this in our college non-majors general biology laboratory course. And then our second part of the project, we're calling a DNA barcoding project. This is actually coming out of a workshop that we attended a few years ago with the DNA Learning Center. And these students are majors biology students. And so perhaps, they're already interested in STEM. Maybe then they can go down the biotechnology pathway. So to give a little bit of overview about this project, so we're looking at some gene inserts on chromosome 6. There are actually 3 different gene inserts that we are studying for this project. They initially were discovered with a group out of Princeton and Oregon State. We have the reference on the bottom of the right of the slide. And we've been able to actually connect with the initial researchers to kind of collaborate. Behavior is, of course, complex for looking at these behaviors. And so we talk about it as being like survival of the friendliest, excuse me. There have been some news articles about this, but really it's more about how attached the dog is to the owner, to other unknown individuals, things like that. So we call it this kind of hyper-social behavior. And that was once again coming from this published journal article out of Princeton and Oregon State. Once again, for our lab project with the students, we are looking at 3 different gene loci, once again on chromosome 6, and seeing if there's a link between genotype and phenotype for our students. And as Misty just mentioned in the chat, we have a video that goes along with this. And Misty, do you want to mention anything about the video? Sorry.
Yeah, the video is the video that we made as part of the project to introduce the project. And we didn't think it would come through particularly well on Zoom. So if you want to use the QR code, then you can see that.
Mm-hmm. And then just to mention, so with this behavior, it's not really a like they're in this hypersocial behavior category or not. It's more of kind of a continuum, like essentially thinking of like, are the dogs more introverted or extroverted? And there'll be some kind of intermediate phenotypes with that as well. So that's just a little bit about the background. And then thinking about the pipeline, so it's really exciting because students get to build their biotech skills doing techniques that they would do very regularly in the lab. So they have an introduction to pipetting. They get to collect a saliva sample from their dog, or if they're not testing their dog, then the dog's owner would be doing this, obviously for safety purposes. They get to extract DNA, run the PCR, run a gel, and then get some experience doing data analysis. And we have done this with, once again, with our college non-majors biology students, also high school like biology students. We've also worked with some middle school students, and they might not do the entire workflow, but they get to do sections of it, get some practice with pipetting, learn about data analysis and things like that. So it's very highly customizable dependent on the level of student.
Okay, so hi, I'm Tracy, and I'm just going to talk to you a little bit about some of the things that are important for the success of this project. And one of the huge things I would say for this project and for our program that we're learning along the way is that really our relationship building with the partners and getting resources and information from other people has been really, really crucial for our success. So obviously Neogen is the partner that the canine care was designed off of, and Neogen also talks in that video. So we want our students to see, you know, these are potential careers that you can go into. This is a local industry where you can get a job and have a career. And then other pieces are our University of Nebraska-Lincoln has an NSF-funded EPSCoR center, and they already have mobile labs. So one of our concerns was, as a community college, we can do this for 3 years, but we're not going to be able to make these. We essentially make kits and deliver them to the high school teachers and provide them with all the consumables. We can't do this for longer than 3 years. We don't have the resources, but our EPSCoR friends at UNL, they already have mobile labs that they ship all over Nebraska. So when we finish this project, they're going to pick up and not only be able to serve our 16 county area, but the whole state of Nebraska. And one thing we're actually realizing this year is it's becoming so popular that the majority of the teachers that have signed up for our workshop this summer are in much rural areas, like further away, that we can't get to. So I think in the future, as far as sustainability, finding those partners that have resources that can extend further has been key for us. BayBeck does high school biotech labs in the San Francisco Bay Area. So we're working with them and they're going to be able to disseminate this in the Bay Area. Mini One is mostly what our equipment is for this lab. It's really accessible for high school classrooms. And then the Linksy is the Lincoln STEM ecosystem. So we're also just trying to partner with as many people as we can in the community to get this out there, because again, our overall goal is, we often hear from our high school teachers, we didn't know that this industry was in our backyard. When they come to Neogen for the workshop, they say, we didn't know this existed. And so our goal is, let's get that information out there. So partnering with those community organizations has been super impactful for our community and for us as well. And then this is sort of a current progress. We can track the numbers in our area. As I mentioned before, we have disseminated this through Innovate Bio nationally. And then also at High Tech, we've already disseminated this throughout the country, which we're not tracking. But so far, we've seen that in our college students, we've hit about 550 students, and then high school students about 1,200 to 1,300 students. So we're pretty happy about that we're reaching so many students in our local area. And my guess is that impact is more in the nation. And I think oh, go ahead, Misty. Sorry. Some things that we're working on, and this may be relevant to this Innovate Bio community if you would like to implement this, is one of the coolest features that we thought of, which in practice on the ground has been a little bit more difficult to implement, is we have a national database. So if you were to do this in your biotech classroom, your biology classroom, whatever classroom, your students can submit their data. It goes to our national database, which the data submission is working well, but the presentation of the database is being optimized by our the people that we're working with, a local company called Don't Panic Labs. So we're still in the process of getting the visual data to be shown for those for the database. And then I mentioned before, our key goal here is to make sure this is sustainable in the future. So as we transition into our last year, we want to really make sure that the teachers can do this independently, and that our system, where we're getting these labs out into the whole state using EPSCoR, we want to make sure that that transition is pretty smooth. And we did have some struggles. We mentioned last year with scaling this up in our non-majors biology lab. We kind of just gave the biology teachers the experiment, and we realized a lot of our non-major biology instructors are more environmental ecology biologists, and we actually needed to go back and train them. So that was just us learning along the way. We made a false assumption and we had to go back and correct. So I think that's one of the lessons we learned from our first ATE grant is we have an idea of how things are going to work, and that's not always true. So we need to go back and get feedback from our people, you know, that we're working with, whatever partners they may be, and we need to adjust our strategies. So I think that's one of the big learning points for us this year. And then, as I mentioned before, we're disseminating this nationally. So if you wanted to come learn about this, if you're at High Tech in July, we'll have a poster there. We are actually doing an ABLE conference in Maryland in June, and that's a conference. If you attend, you can actually do the 3-hour lab with us. Like, it's a 3-hour session where we're doing the whole lab, and you would get to analyze the data. Here's just a snapshot of what that data looks like for a class. There's, you know, several dogs that were swabbed, and you can look at the gel on the right. This is different gene locations. So in the paper, the original paper, we're naming them after those gene locations. There's 6.66, 6.7, and 6.6. And you can see the lower band, that's about 250. That is wild type, or what they associate with wolves, the behavior that is not as attached to a human, more independent. And then the higher band at 500, and sometimes 550 at some of these at 6.7, that is associated with more attachment to the person. So, and then off to the right, you can see the hypersociability score. So currently, based on the paper, we give students a dog survey of 5 questions, and they answer those questions to get a phenotype score based on that survey. And so this is a current data set that we have. And then, of course, when that national database is fully corrected, you'll be able to see all the results that have been submitted for those gene loci.
And I would just add there that most of the time the high school students are they're just going to do just one gene loci, and we've been having them do 6.6, and other more advanced classes, maybe college level biotech classes, for example, will do all 3.
And then here's all of our contact information, and also a QR code that has all the resources for all the curriculum. So all the protocols, prep material, that video that Misty mentioned, and the link to the database.
Yeah, so this is the link to the Google Drive that has all that stuff that you are more than welcome to go and see what you can use.
Yeah. And then the other thing is, if you go through this and you have questions, feel free to reach out to us. And I can give you, if there's something that's missing or you have more in-depth questions, we can meet with you via Zoom, you know, we can answer via email, whatever works best for you and your team, we're available.
Sure. Yeah, so if there are questions.
We do have questions.
But we're going to wait and ask.
I realize that. So I just wanted to tell you, we do have questions.
And what we'll do is at the end of the second talk, then we'll open it up to all the questions, if that's okay. We appreciate your presentation. Thank you again, Misty, Tracy, and Gabrielle. Please join me in welcoming our second talk speakers from Johnston Community College in Smithville, North Carolina, Dr. Brian Worley, who is a PI. Joining Brian is career coach Julie Griffith, as well as evaluators John Cosgrove and Maggie Cosgrove. Their presentation is titled BioBlend 1.0, Using Self-Assessment to Stay on Track. They will share grant background information and a self-assessment tool created by the Cosgroves to monitor grant progress and evaluate partnerships, activities, challenges, and actions taken to address the challenges and outcomes. Welcome.
Thank you, Dion. I'm Brian Worley. Before we get started, I'd like to make sure my team turns their mics on so that when it gets their turn to talk, they're ready to go. So Julie, if you advance us to the next slide, please. Okay. Again, I'm Brian Worley. I was the principal investigator on this grant. My job title at the college is I'm the Associate Vice President of Business, Industry, Logistics, and Transportation. And the part that my departments played in this grant was the applied engineering side of it. As we heard before, our career coach, Julie Griffin, was a huge part of this grant and making us be very successful. And what really made it successful was John and Maggie Cosgrove, who were our evaluators during the whole grant process. Julie, next slide.
Oops. Excuse me 1 second. Here we go.
There you go. Thank you. So I didn't know that we we're going to have a guest on here today that I wasn't expecting to see. And I want to give a shout out to Miss Elaine Johnson, because I believe that if it wasn't for her, whenever we were in Mentor Connect in 2018 and her leadership and helping us develop our grant and receiving the funding that you can see here for $283,000, we would not have been able to continue to BioBlend 2.0 or the other grants that we've submitted to NSF ATE. So I want to thank you for your mentorship and leadership way back in 2018 to get us where we're at now. So thank you. But that being said, we started out with a very simple idea of being able to integrate some biotechnology classes into our applied engineering program. And again, we were funded. And through that, we were able to grow that, continue that grant into BioBlend 2.0, where we were able to put into that grant a neurodiversity piece where we work with autistic students and give them the opportunity to get some of these high-paying jobs in our pharmaceutical industry here in North Carolina. So it's been a win-win for us all the way through. Next slide, please, Julie.
Give me 1 second. Oops. Hold on 1 second here. There we go.
All right. So we had a couple of goals. The first goal was to create and implement a unique certificate where we, as I said, we took applied engineering classes and the bioprocess classes, and I'll show you more in depth what these classes are on the next slide. But then we also created a multi-skilled talent pipeline from the college to industry. And then our third goal was to do some hands-on drug simulation in a manufacturing environment. And Julie's going to talk about that later, but that was Delta V. And we're super excited about that. But what you're looking at right here is the original classes we started off with, which was ELC 117, MET 110, BPM 110, and PTC 110. Okay. So here you can see a breakdown of what those classes were. Again, the bioprocess practices was a 5-credit class. The industrial environment, that's where the students get their aseptic training. And then the MNT 110 is intro to maintenance, and the ELC is motors and controls. Okay, so you're probably saying, well, okay, what was the purpose of this? Well, our pharmaceutical partners said, you know, what we really need is we need technicians or people that come to work for us that can do some basic troubleshooting. And that way, whenever the technicians get there to do the repair, they can kind of help them real quickly identify what the issue is and get back up and going. That was the win side for the the bioprocess students. They get those maintenance and electrical classes. The win for the applied engineering students was they learned how to work in those environments. They learned how to do aseptic, how to be clean when they take their tools into those areas and not contaminate everything. So it was a real good win. It was a great blend of these 4 classes to create this certificate for our students. And at this point, I'm going to turn it over to Julie, and she's going to elaborate a little bit more into what all we did.
Hello, everybody. Glad to be here from sunny North Carolina. As Brian mentioned earlier, I'm Julie Griffin. I'm the career coach. I was hired at the very end of year 1, going into year 2 as a career coach to really support the activities and outcomes of Objective 2, which was to create a multi-skilled talent pipeline from college to industry. I supported 25 plus students. Really, we had 25 students and 2 cohorts come through the program, and supported them in resume development, mock interview skills, facilitating internships, coordinating employment interviews. And then also during this time is where we incorporated speed networking into the program, which that presentation will be on April 26th. Our third objective for this grant, BioBlend 1.0, is to provide a hands-on education in a simulated drug manufacturing environment. And the picture that you're looking at is in our simulated manufacturing work environment, pharmaceutical manufacturing work environment. What you're looking at is a performance learning platform that Emerson has, that Emerson developed. It's got all the bells and whistles to provide simulation of what it would be like in a pharmaceutical manufacturing company. It connects to water, has valves, piping, and has the Delta V interface as well. As Brian mentioned, the third main objective for this grant was to develop that Delta V certificate. And we were, in fact, the first community college in North Carolina to develop that Delta V certificate. Delta V is a distributed control system that our major biopharmaceutical manufacturing partners, Grifols and Novo Nordisk, use in their operations, as well as companies outside of biopharmaceutical manufacturing. Students participate in 3-day Delta V training course, and then they come out with a certificate. We also, as part of the second grant that Brian mentioned, our expansion grant, BioBlend 2.0, have purchased a clean in place skid, and we'll be connecting Delta V to that so students can actually operate a piece of equipment using Delta V. Just to share a little bit about our outcomes, again, we had 25 students come through the two cohorts. 22 of those students graduated with associate's degrees. 23 obtained the BioBlend certificates that Brian mentioned. 20 individuals participated in internships. We actually had 4 students move into employment just before internships launched. And then we have had 18 of those students obtain positions in biopharmaceutical manufacturing and 25 Delta V certificates obtained. What really helped us stay on track was utilizing the self-assessment tool provided by our evaluators, John and Maggie Cosgrove. This tool helped us monitor our own progress, our relationships. We rated our relationships with our external partners, identified challenges and accomplishments, and it really helped us to stay on track. We actually nominated our evaluation report for an award via Evaluate, and the Cosgroves and our award won. So we're really excited about that and excited that the Cosgroves are here today to share a little bit more with you about the self-assessment tool. And John and Maggie, if you're here, you ready to take it away?
We're here. So first of all, I have to say this group was really, we're evaluators. We work with many colleges and universities across the country. This was a great group to work with because right from the beginning, the college was excited about getting this grant. And, lots of times colleges are excited about getting this grant, but sometimes it's the grant team that's excited. But in this case, the college was excited. And it was not just the grant, it's a great grant team, but it was beyond that they were excited about it. And it's our goal in evaluation to help all the grantees that we work with get better, be the best that they can be. And at this college, Johnston Community College, they really wanted to do that. So that's what we mean by continuous improvement, is this goal to get better. And we think about evaluation across all these different parts of the grant, how the grant processes are in place, how the implementation is going. Are you doing what you said you were going to do? Are you making progress? What do your outcomes look like? And from the very beginning, are you thinking about sustainability? So go to the next slide, Julie, please. So in this grant, the college was trying out some new things. Of course, curriculum was a big deal for them. This, when you blend 2 departments together, that sounds easy, but it's not always easy to do. It's 2 different languages or maybe more that are being spoken there. Career coaching was something new to the college, and the innovation in partnerships was new to the college too. So we worked with the grant team to take a tool that we use across most of our projects, but to sort of customize it, to what do you care about? What do you want to keep track of? And let's follow that in a systematic way to determine how you're doing. So if you go to the next slide, we can kind of see an example. So the partners were really important in this grant. So one of the very first things we did was John and I suggested some possible roles partners might take along the course of the grant. Of course, much of this we got from the grant proposal, but also through conversations with the grant team. And then they came up with, these are the ones that we care. And there's actually more than these, but these were the ones that they cared about. So it seems like it was twice a year in the beginning, and then probably twice a year throughout the course of the grant. The grant team evaluated each of their partners along these goals. So if you go to the next slide, Julie, you can see that we actually had the college thought about, well, what kind of partners are we going to have? Is our employer partners? High schools were a big deal here. Within the college, internal college, and then they worked with professional organizations outside of employers or the college. So they identified what kind of partner it was. And then it was important to, when you write the grant, you have these hopes that your partners they promise, right? You have hopes that they're going to do what they say. So each time the college did their self-assessment, they said, well, are they meeting? Are they what we expected, or are they just blowing us out of the water, or in one way or the other? So they looked at that each time. And then across those roles that we showed you in the previous slide, they just did a 1, 2, 3. Is it low involvement? Is it medium? Or is it high? And that was a good way for them to look at their partners and say, why is it that this one particular high school isn't doing so well? And also the conversations that the grant team had when maybe Julie would say, I think this high school is a 1, and someone else on the team would say, oh no, they're really involved. So it's a great time to get conversations about topics that you might not talk about on the regular course of in your grant team meetings, if you will. And then in the next slide, Julie. Oh, okay. So for each of the grant goals, the college had activities, outcomes, outputs, objectives, maybe. And so we agreed upon this scale for them to self-assess where they were for each of these items. And if you go to the next slide, I think this will make a little bit more sense. So here, but for example, the first goal, here's the activities, the outputs, and the outcomes. Each time the grant team got together and said, where are we in terms of implementing this? And along with that, they would ask themselves questions that what do we call those, Jen? Questions to enhance learning. Mm-hmm. And so they self-assessed each of these, and sometimes, like at the beginning of the grant, things are always at the planning stage. But if it's still at the planning stage at the end of year 2, you got to question yourself, we forgot we promised to do that. So this is a way that you don't overlook anything. So if you go to the next slide, these are the questions that really, we ask them to just free-form answer this. And we always said it doesn't need to be a beautiful paragraph. It could just be bullet points, but tell us, what did you do? What was challenging? And what did you learn? For each goal, we ask this, and then overview, sort of at a higher level across the grant, what are your accomplishments and what are your challenges? And so twice a year, the team would get together, answer, ask themselves these questions, and then give the answers to us. And then we would have conversations with the team. And I think those were the conversations that where it really gave the team a time not to actually work on the grant, but to think about the grant and to reflect on what they were doing and to have ideas for moving forward.
Maggie, if I can, I'd like to say something right here.
Sure.
This evaluation tool was so beneficial to us because a couple slides back, you saw where we listed the classes that we originally started with. Well, through this assessment tool and what did we learn, the challenges, we found out that the ELC class that we had originally started out with and we thought was going to be a huge benefit to the students was not a huge benefit for the students. It was actually more difficult than we intended it for it to be for the students coming from the bioprocess. So we actually changed in the second year to automation class where the students, because all of the students have to work with cobots in the pharmaceutical industry. So we changed the class from electrical to robotics, and that seemed to be a huge hit. The students loved it. So by, addressing what we wanted to do to make the students successful through this assessment tool, it really, really helped us get the students to where they needed to be functional in the industry. Sorry.
And I would also say, that's great to hear, but I would also say another thing that the tool does is it sort of provides this bookmark, if you will. So if there are any changes in staff along the way, it's a great way to say, oh, here's a little notebook of where we've been at a high level rather than all the little pieces that I've been doing to work on the curriculum or the myriad of things that Julie did and started for the first time at the college as the career coach or the individual work with employers. This was a way to sort of note those things. Maybe like an organizational tool, if you will. A planner, let's call it. And I think that might be, is that the last slide, Julie? Did we end with accomplished? Oh, no. Oh my gosh, no. So this is how we view this self-assessment process. And as evaluators, what John and I do is, so the colleges self-assess, and then we don't just take their word for it. We triangulate that data with interviews with employers, interviews with student records, with interviews with other people within the college to make sure, and we have campus site visits so that we can say, yeah, they've got a clue on it, or we will say to them, you guys think you're doing poorly here, but people are telling us, no, you're doing well. So even though their self-assessment is important, we feel the need to sort of confirm it. Okay. Thank you.
So are there any questions? I have questions for both.
You want me to go ahead and stop sharing?
Well, don't stop sharing. Can I ask my
Oh,
question on your slide?
Too late.
then people have other questions. Go back, because I didn't understand
All
one
right, hold
slide.
on 1 second.
And then I see Travis has questions too, but I'll take the upper hand of asking my one question. Go back. I didn't understand one slide that you had on the evaluation. So go back. Back. Keep going. Keep going. Keep going. Keep going. Keep going. That one. So type of partner, employer, scale for the degree partner meets expectations well below our expectation.
Mm-hmm.
What does
So,
that?
So what does that mean? Well, so we have these responses for the college. And basically this is a change. Chance for them to discuss how well we just created these columns of things that we look at. But with regard to the scale to which partners meet expectations, we found that many times colleges enter a grant and they believe that this partner is going to be great. Or sometimes what happens, and this happens actually pretty often, is a college is working with a partner and then the person at that partner is transferred to another country or they retire or whatever. And the new person comes on and says, you know what, I don't really care about that project at all. And so that partner who you thought was going to be really important is now functioning well below what you expected when you wrote the grant. And that means you got to get out there and find someone else to do whatever it was that you expected that partner to do. So this is a way for the college to think about the roles the partner plays. Not only are they doing as well as they hoped, but you know, within the various things that they hoped they would do, how well are they doing?
Great.
Well, and it's important, so it's important also, if you've got, kind of a set of outcomes, that are linked to the partnerships. Well, it's important to know really if, the partnership that is active and that can meet in expectations, right? Because if you don't know that, you might jump to the conclusion that says, oh, the partnership was ill-defined or something, right? When it had nothing to do with the structure of the partnership, but it had everything to do with the fact that whoever the partner was, either organizationally or at an individual kind of level, they weren't, up to the expectations. So in effect, no outcomes were occurring, right? So it gives kind of the grantee and the evaluator, the opportunity to really kind of connect this notion of implementation and outcomes so that you don't jump to conclusions about stuff that you don't have information on to have an understanding of the implementation of the partnership.
Well, I want to add to this part of the self-assessment tool, our external partnerships. We're also rating our partners as individuals that hire our students, individuals that contribute to curriculum, building some of the curriculum. So there's other categories on this as well that helps us get to the meeting our expectation, low, medium, high involvement. And I will say too that I go back to our letters of support a lot as well. What did we say at the beginning? What did our partners say at the beginning that they would support in our efforts if we were awarded this grant too?
Yeah. Travis.
Hi, so I'm at a community college in Washington State. I'm just now starting to think about developing biotech programs and at the beginning stages of reaching out to industry partners and so on and other community partners. So I have questions for both the Johnston Community College team and the Southeast team, and really for everybody who has more experience than I do. So for the Johnston Community College team, when did you contract with the Cosgrove evaluators? At what point in your program project development did you start working on the self-assessment, which seems like it should be something that I should already start doing right now? And then for the Southeast team, hi Tracy, I met you in New Hampshire this past summer. You have a certificate and the certificate, and then all the way up to associate's degree. And this question really is for everybody. Just starting out and thinking about engaging industry partners. It's my I get the impression that they don't really see how community college students could fit into their workforce. And I'm not sure if that's true or not broadly, but I kind of get that impression here. How did you engage industry partners and talk about the value of your students in their programs?
So this is Maggie Cosgrove. I'll start. So Johnston Community College reached out to us in the grant writing stage. They needed an evaluation plan. And so they gave us their grant in rough draft. We had several conversations, and then we wrote the evaluation plan and they submitted it. And then we said in the evaluation plan we were going to do something like this, but it wasn't, it wasn't a pipe dream, but it wasn't all spelled out. And after the award, and then once we got the contract, then we sat down. I think we passed this tool back and forth 3 or 4 times before, it was like, okay. And then, you know what, the tool got, I don't know, Brian and Julie, I think the tool got edited, changed up a little bit, as we learn things. Oh, we need to care about this more than that. So it was an evolution. But you're right, it has to start early.
Yeah, the very beginning.
Mhm.
Now, Travis, to answer your question about our pharmaceutical partners, we've got two giant companies where we live in North Carolina. And I think, Julie, correct me on this if I'm wrong, but I think there's like 75 pharmaceutical companies within a 50-mile radius of where we're at. So we're just inundated with pharmaceutical companies here in North Carolina. So they value what we're doing. Our biotech students, unless it's changed, they all get an interview with the companies locally. So they really buy into what we're doing, and they really appreciate. I mean, they're the reason we wrote the grant. They're the ones who came to us, and they wanted us to create, before we even knew we could do a NSF ATE grant, they wanted us to just try to do it ourselves. And then we saw this as an opportunity. And it worked out very well, because otherwise we would have never been able to get into the Delta V arena because of the cost.
Hey, Travis, so we have a industry meeting April 17th and 18th here in Seattle. And you should probably come, because I'm sure, ABSCI, the company in your area, they just got $86 million and they'll be there talking. So it's a good chance to come talk to them.
That's the Life Science Washington meeting?
Yeah, Life Science Washington.
I'll be there. Thanks.
I can also add that as we've started our program, our industry has actually been fairly open to hiring people with a 2-year degree or less. The workforce demands here are high enough that they just have trouble finding anyone who's willing to work. And so if they already have some training, they appreciate that. And they also appreciate that they can give us direct input about what we teach, and that we can be responsive.
Other questions? So I have a question about the dogs. So what did you find out about the dogs? I mean, this sounds so fascinating. I'm sure people were interested in doing this because they want to know about their dogs.
Yeah, everybody does love their dog, and we've found that more and more. Everybody wants to give you their dog sample, even if they don't get to run the whole experiment. But our industry partner was the one who had been already looking at the research to incorporate this into one of their veterinary tests. So tests that they offer to veterinarians to offer to their pet owners, because in addition to breed, people want to know more about the health traits of their dog. So they were the ones who directed us to this might be a good thing, and they'd already been working on doing the PCR. We had to adapt it to be able to work in a classroom setting for sure. And actually that was a very short timeline. And I'll give Tracy a shout out for really crunching that out, because we had a workshop coming up with high school teachers, and the company was hosting it, and they were basically going to take people on tours, these high school teachers, and we're like, but they need something hands-on to take to their classroom. And so we said, let's try this. And so working together, we were able to at least get a start on it, which led us to then filling in all the details over time.
And the correlation between hypersocial and the site was
basically the hypothesis is that the more insertions the dog has at those 3 gene loci, the more hypersocial the dog would be. Currently, the data that we're getting based upon the aggregate score of the survey data is showing an inverse correlation, but it could be the survey that people want their dog to be hypersocial, and so they're answering the survey questions with higher numbers than perhaps they would if it was completely unbiased. And so we have tried to add a behavior test where the dog is you're someone sitting in a chair, and you're then monitoring how much time the dog spends close to an unfamiliar person and then a familiar person. So the
Uh-huh.
unfamiliar person part is tricky because a lot of times the students are trying to collect their sample from a dog at home. They don't just have someone unfamiliar to the dog around. And some people are uncomfortable with that idea of like bringing someone unfamiliar to their dog to do a test with as well. So that was the test that the people at Oregon State had done in their research, and they use that a lot. But they're having people come to their like center for research and then monitoring that whole test. And we're trying to do this as a citizen science project. So that part has been hard to get buy-in to do, which would provide more accurate data.
And you're going to be able to track within the life of the grant. Are you going to be able to track the freshman, sophomore?
Not from
possibly?
high school, no, because our grant will end by the time that they're seniors in high
But
school.
what would it take you to do that? Look at
Well,
money.
well,
What will it take you to do this? Because you realize how valuable this is. I got into a previous conversation about, yes, engagement in 9th grade is great, but you have to show that you actually get students. That'll be the first thing a program officer asks.
You can
Yeah.
apply for a supplement. You can
yeah, we
extend
might, maybe
your
we'll
grant.
have to do that. Yeah, I don't know that, but like we don't have data on like which exact students have completed the project. We just have information on the teachers mostly at the high school level. We know
Oh,
how many students are in their class. We don't have like every student's name.
Oh yeah, but you can go back and get.
no, but you get if you get students who are applying to their program, your program, right? They start
And
your
then
program,
ask them
you can have a
why.
survey and
Yeah.
ask.
Right.
For sure, we can certainly ask them once they come to us if they had
Yes.
exposure to it previously, for sure.
Yeah.
Yeah.
Do you survey the students at all, just within the class, attitudes towards biotech prior to the class and are the sessions and after?
We do that with our college students, but we did not go to the effort of, we didn't know exactly which teachers were going to be participating and which school districts they would be from. And so we did not attempt to get survey data from the high school students.
Yeah.
So we have it from college students though.
Yeah, just asking everyone who enters your program, how did you get here?
Yeah, we can certainly do that.
And if you want advice about applying for a supplement, just get in touch with me.
Okay, because
Yeah,
you know,
you should do that. Or, the center would gladly help with this because that is the big question.
Yep.
I was
So
going to add
serious.
that Brian and I, we didn't mention it, but with BioBlend 1.0, we did ask for a no-cost extension and then a small supplement to finish out a second cohort of students.
yeah, so we do have to decide, well, a couple of things. This of course is the second year, so we need to decide if we want to apply for some way to expand this even further through ATE. Or the other thing is with the supplement, we've actually had more people want to participate than we had budgeted for. So we may need to ask for a supplement for that as well.
You can do it. It's not that hard.
Okay.
Sandy,
Do both.
volunteer to help in this. Go
Okay.
for it.
I would echo, it was a simple process. I mean, obviously there's some, you need to justify the needs and show the budget, but
Right.
it's only a program officer review if it's 20% of your annual award or less,
Okay.
instead of a full panel review.
Okay. Great.
And it's short. It's not 15 pages.
That's
And-
No.
Oh, and the survey. Dion has posted a link to a survey. We really will appreciate people filling out because that helps guide our future work and the series. And so please don't leave without make sure you click that and fill it out.
And
Thank
again,
you.
Thank you to all of our Project Talk speakers today. And as Sandy mentioned, yes, we would appreciate you completing the survey, which will help us in planning for future events. And if you would like to look for additional information on these project talks, you can go to the Innovate Bio website, which I posted at the beginning of the chat. And you can also register for future ATE project talks. Again, thank you so much for attending. And those of you who have spring break coming up, we wish you a safe and peaceful spring break. Innovate Bio team are those that are included in spring break, but we all know that Linnea will continue working.
That is so true. I may be in Colorado, but I will continue working.
But thank you all to everyone again, and we appreciate your attendance, and let us know if we can help you in the
Yes.
future. Thank you and have a great day.
Thank you.
all.